Welcome to the Hatchery online learning resource for the FP7 Nephrops project. In this module we will learn about broodstock biology, handling and husbandry procedures. The term buried usually refers to sexually mature females that are carrying fertilized eggs on their abdomen. These are extruded during the winter, usually from July through to October. The eggs go through several development stages which can be defined by visual assessment of the changing colour of the embryos. The fecundity of the females also varies with location, with around 900 to 60,000 eggs extruded per female. A more detailed and definitive description of the egg stages can be acquired under the microscope by gently removing several eggs with some tweezers and analysing the amount of yolk left. The eggs change from dark green in the early stages, as seen in the top left, to a lighter olive colour, with a very obvious eye spot just before hatching, as seen in the bottom right-hand picture. This final stage usually occurs in the months April to July, but this depends on the location and the temperature of the water. Broodstock should be brought in from February onwards throughout the spring and early summer. This is preferable to overwintering, as from these animals there are fewer released larvae and reduced survival to later stages. The procurement of broodstock needs to be local to remove several stressors during transportation to the hatchery facilities. Three main methods have been used to procure the broodstock. These are directly with the hatchery operators in full control of capture and transport to the hatchery through the creel fishers themselves or through a shellfish merchant. The table illustrates the pros and cons for the different procurement methods. Procurement from the creel fishers is recommended as it involves lower costs in time and transport. Procurement through a shellfish merchant will usually involve using your own transport, and extra care needs to be taken in handling these animals, which may have variable provenance. There are several ways to transport the broodstock, and the most appropriate method will depend on cost and the distance between the supplier and the hatchery. Damp transportation can be used successfully during a journey time of less than 2 to 3 hours. This uses damp tissue paper, a towel or seaweed on top of the tray, or dispersed between individuals and then chilled down with the use of ice blocks. Always make sure the ice blocks don't come into direct contact with the brood, as this may cause potential damage. Tube trays immersed in cold seawater are recommended, as they are easily available and a practical way to transport the broodstock. It has the advantage of keeping the individuals separate and at a relatively high density. Otherwise, the females can be placed communally in a single tank but may need to have their chelae banded. Vivier systems are used commercially and are preferred for journey times longer than 3 hours. The broodstock are submerged, the water is recirculated, and the temperature, ammonia and oxygen levels are regulated. Both transport methods can be achieved cheaply. Illustrated here is the system set up using basic equipment that we used during this project to transport over 300 individuals at a time over several hundred miles. Please see the Nephrops hatchery manual for more details. On arrival at the hatchery, the broodstock should be put into the holding tanks as soon as possible. Leaving the individuals out of water for prolonged periods of time will cause increased stress and mortality. To remove the brood from the tube trays, you need to place the tray upside down and remove the bottom to allow access to the brood from the tail side. Alternatively, use a pipette to allow the brood to grab the chelae and pull the individual out. To check the condition or egg stages of individuals, it may be necessary to handle them. The time out of water should be minimized, light conditions dim if possible, and temperature low. Illustrated here is how to correctly hold the nephrops. Hold the nephrops on the dorsal side of the carapace, but caution is needed as the chelae can reach around to the back. Hold the animal between your thumb and first finger and place it either side of the carapace just below where the first 2 chelae emerge. Allow the telson to push upon your palm, as this will stop it tail flipping and reduce egg loss in buried females. When returning the animals to the water, always place them in tail first and upright. This will allow any air bubbles caught in the gills to escape. The broodstock can be held in the holding tanks either communally or individually. Preference should be towards housing to reduce aggressive interactions and lower stress levels during embryo development and hatching. It also allows easy monitoring of the specific individuals. The cell matrix illustrated on the right comprises solid walls and perforated floors to allow for water exchange and circulation. Other variants include larger perforated cell walls that allow larvae through to be collected near the tank outflow. All tank outflows should have a mesh filter of around 1 to 2 mm to prevent hatched larvae from escaping. Some of the important water parameters are shown on this slide. It's important to note that the broodstock should be kept in an undisturbed area. Having adequate water filtration, flow-through, sterilisation and cleaning procedures are important for hatching success. Water flow within the holding tanks should be high enough to exchange water and maintain dissolved oxygen levels and maintain the required temperature, but low enough as to not damage the hatched larvae or the broodstock. This may need experimentation with your specific tank design and setup, but it's typically quite low. Regular water testing is essential to maintain adequate amounts of dissolved oxygen in the tank. The water temperature within the holding tanks should mimic that of where they have been caught, although the temperature control will impact upon development. Therefore, you can prolong the season with cooler temperatures or speed up the onset of larvae hatching by increasing the temperature, but the rate of change should be no more than 0.5 degrees per day. It's also important to keep the broodstock in a low light environment to mimic the low light levels experienced in the wild and on a photo period that reflects that of the natural environment for the time of year and location. Most larvae will hatch at the onset of dusk into darkness. You can also use red light filters, although this makes the larvae harder to see. The broodstock can be fed a variety of frozen or fresh seafood. The most convenient is frozen prawn tails, which can simply be defrosted and fed, one tail per individual, once or twice a week. Fish or mussel tends to leach and break apart and is therefore not recommended. Any uneaten food should be removed after 24 hours. Cells and holding tanks should be cleaned regularly to maintain low bacteria levels from the buildup of uneaten food, feces and dropped eggs. Filters should be checked and cleaned daily to reduce the potential of an overflow from a blocked outflow and also reduce bacteria levels. Egg development should be checked out once every 3 or 4 weeks so as not to cause unnecessary stress due to repeated handling, which will have a negative impact on larval hatching. There are several ways to do this. One is to use a dental mirror to view the colour of the eggs without directly handling the female. Another is to take the female out and inspect the embryo. This can be done directly with your hand as shown previously or with the use of a plastic pipe. The pipe can be used to scoop the female from the rear as seen in the right hand picture. It's important to record embryo development in order to prepare for larval hatching an estimated number of larvae. As seen in the left-hand picture, these embryos are in the final stages of development and will hatch within the next few days. Females that are no longer carrying embryos or have proceeded to molt can be removed from the broodstock holding tanks. These females can be released depending on local regulations. There may be some instances where females remain buried and do not release larvae for the entire season. Hatching occurs over 3 to 4 nights for a particular female. It's more practical to collect all the hatched larvae the following morning, although earlier collection and feeding straight from hatching would be beneficial to larval survival. However, this may disturb other females in the hatching process. Placing live food in the holding tanks could be beneficial for newly hatched larvae to feed upon during the first few hours and may enhance later survival. The larvae are extremely delicate and must be handled with care. The larvae can only be transferred using water-to-water techniques. Using a net would damage and kill the larvae. Using a beaker or a gravy baster is the best method to collect and also to observe the larvae. Once the larvae are collected, they are counted in a collection jug or bucket. Make sure the water is of similar temperature to the tank that they have been taken from and that the larvae do not remain in the jug for long periods of time, as the oxygen levels will decline and the temperature will increase. Also, be mindful of the density of larvae within the collecting jug, as the larvae can turn cannibalistic. Once the larvae are collected and counted, they can be transferred to larval rearing tanks. Transporting the larvae in the collection jugs is satisfactory for short distances. Care needs to be given that the larvae are not exposed to sudden changes in temperature and light intensity. That's the end of the broodstock module. In the next module, there will be a discussion on how to raise the larvae to post-larval stage 1.